Good afternoon. We are waiting for. Okay, now we can start. Thank you again and again for your participation. It's time to start our green biotechnology section. And I'm not sure, but it looks like the first speaker is absent, but just in case, I will check. Is Zakiriaeva Saidahton with us? No. Then our next speaker is Varona Valentina.
I'm here.
Okay, you're welcome. Can you share your screen, please? Yes, thank you.
Can you see my presentation?
Yes, we can see it.
Okay. Good day, everyone, and thank you, the organizer, for this conference. I'm glad to see you all. And how we know, the pollutant caused by plastic is one of the greatest threats to the environment, and this is because of their non-degradability, persistent, and recalcitrant nature. This contamination can result to severe health and ecological prestige to humans. Recently, the biodegradation of plastic and plant bacteria or fungi had increased from months due to their efficiency, effectiveness, environmental friendly, and sustainable development. In accordance with the above, then our study was to optimize the condition for creating a microbial consortium isolated from the soil, which participates in the bioconversion of non-recyclable plastic by using the methodology of accumulation culture with the environment of microorganisms that colonize the LPC surface. The microbial complexes that are involved in the decomposition of nano-recyclable plastic created in the polluted soil collected from landfill situated close to Slobozia Duska village. Consortium of these microorganisms isolated from soil were treated with LDP under aerobic conditions on liquid media. The microbial complexes that are involved in the composition of nano-recycled plastic, as I said, consortia of microorganisms isolated from soil LDP treated under aerobic conditions with two mineral types, liquid salt media, MS2 and MS4, to obtain enrichment culture. MS2 media is favoring with the growth of macromycetes with pH 5.5, and with bacteria we adjusted to 6.5. These two mineral media are different in salt content and nitrogen, phosphorus, and carbon ratio. And at the initial stage of obtaining consortia in the cultivation media, LDP was added in the form of granules, an amount of 1 gram. The systematic group microorganisms present in the consortia were observed during the experiment by inoculation as solid selective media like MS2 agar medium, Kinetic media, and nutrient agar. The result demonstrates that after 100 days of cultivation, the microorganisms in the consortia attain their viability. The titer begins from in the range 7 to 26, 10 to the 6th power. If we analyze the consortia of microorganisms obtained, which included mycelia, fungi, and bacteria, we observe the mostly predominant is fungi. For the most part representative of the Trichoderma species, Penicillium species, and Fusarium, while only one bacteria we have, Pseudomonas, which was identified in 7. And in the conclusion, 3 genus of micromycetes and of bacteria was isolated, described, and determined, which can grow in mineral media in presence of LDPE. And this LDPE can this organized can use the plastic like a potential source of carbon on an energy. And thank you for your attention.
Thank you. Are there any questions? I have a question. The conclusion that the consortia can use LDPE as a source of carbon comes from the fact that the media did not contain other sources of carbon. Is it correct?
Use glucose.
Yeah, but it was introduced to start the growth of the biomass. So I'll put it in a different way. How do you know that your consortia can degrade LDPE?
The second part of our result, we put some form of granulated plastic.
Okay.
And then use this like source carbon and energy. We saw the result with some oxidized information, and we see this result.
Okay. Are there more questions? Thank you very much. Our next speaker is Tengaich Aurelia, and she is to present us a report on entomopathogenic biopesticides. Okay. Яичный держа, да? Где?
Экран.
Есть, да?
Thank you very much. I want to present my work. I represent a group of scientists of the Institute of Genetic Physiology and Protection Plant. Așa. Cercetările au fost realizate în cadrul proiectului programului de stat și a proiectului bilateral Moldova-Belarus. Proposed consists in possibility of entomopathogenic strains Bacillus thuringiensis courstachii and Bacillus thuringiensis turgidis application for spraying. Objective, biological control of harmful organisms using the complex based of para-aminobenozoic acid derivat in a microorganism. Nivelul actual de protecție a plantei nu rezolvă toate problemele producției de plante, deoarece pesticidele nu sunt capabile să protejeze plantele de stresul abiotic. Între timp, pierderile din cauza Factorul de stres sunt estimate la 51-82%. Potrivit afirmațiilor, volumul producției agricole în Republica Moldova se va reduce cu 25%. La summitul mondial din Glasgow s-a menționat de nenumărate ori cele mai sărace țări vor suferi și deja suferă cel mai mult din cauza schimbărilor climatice. Spre deosebire de principalele țări poluante, acestea nu au resurse pentru de a se adapta rapid, dar cu atât mai mult aceste țări ar trebui să gândească la problema dată cât mai devreme. Moldova face parte din categoria acestor țări. Încălzirea treptată, dar stabilă, afectează deja Republica Moldova ca și în alte țări de pe glob. Menționăm că, potrivit datelor Institutului pentru Resurse Mondiale, Moldova este acum printre liderii țării cu un risc sporit de secetă. Conform datelor meteorologilor, se așteaptă până anul 2050 ridicarea sumei temperaturilor cu plus 2,6 grade și scăderea precipitațiilor. Ca rezultat, acest fapt influențează negativ asupra imunității plantei. În acest caz, ar trebui să intervină tehnologiile noi. Utilizarea în comun a preparatelor entomopatogene și derivațiilor para-aminobenzoici ar favoriza imunitatea adaptivă a plantei împotriva stresului abiotic, cât și la expuneri repetate. Devine tot mai evident necesitatea utilizării bacteriilor entomopatogene din genul Bacillus și produselor în metabolismul lor. Una dintre cele mai eficiente și utilizate produse pe scară largă în controlul insectelor sunt bazate pe specia Bacillus. În prezent există peste 400 de formulări pe baza de BT care au fost înregistrate pe piața mondială și este una dintre cele mai promițătoare alternative produselor chimici. Derivația acidului para-aminobenzoic constituie o componentă importantă în sistemul de reglare al plantelor și sunt capabile de a stimula multe procese vitale. În rezultat, acestea conduc la sporirea rezistenței inespecifice a plantei față de factorii nefavorabili de mediul înconjurător. La crearea condițiilor optime se intensifică efectul pozitiv asupra plantelor. Bacteriile Bacillus thuringiensis sunt cele mai adesea folosite ca agenți de control biologic al insectelor dăunătoare. Bacillus thuringiensis, bacterii formatoare de spori, în timpul sporulării produce și o proteină cristalică parasporală care are un efect toxic asupra insectelor. Această proprietate a condus la utilizarea pe scară largă a BT ca baza a preparatelor entomopatogene ecologice ca alternativă la pesticidele chimice sintetice. Anul acesta au fost obținute cu partenerii noștri din Belarus probele de laborator, unde au fost prezentate 10 probe și noi le-am testat în complex cu derivații acidului para-aminobenzoic și BT. În acest scop a fost examinat in vitro efectul concentrațiilor recomandate și jumătate de acid para-aminobenzoic 10 la minus a 2, 10 minus a 4, 10 a 6 mol pe litru asupra coloniilor de bacterii. Bacteriile au fost cultivate în mediul nutritiv mineral lichid timp de 48 de ore la temperatura de 29 de grade, cu titrul la 10 la 9. Suspenzia a fost inoculată pe mediul nutritiv, agarizat în vase petri. După ce cultivarea bacteriei au crescut timp de 24 de ore pe suprafața acestora, au fost plasate discuri sterile înmuiate emulsiile substanțelor cu biologic active. După o săptămână de incubare s-a înregistrat interacțiunea concentrațiilor studiate de acest paraaminobenzoic cu cultura bacteriană. Zonele de inhibare a creșterii bacteriene nu au fost stabilite, ceea ce ne permite să concluzionăm că este posibil să combine soluțiile de lucru ale bioregulatorilor cu suspenzile tulpinilor bacteriene. Rezultate similare au fost raportate pentru amestecul de BT cu pesticide sume alfa, reagente de deces, pseudomonas cu pesticidele ridomil, quadristraxil și specia bacillus. Așa. Aici avem variantele, s-a administrat cu 0,3 suspenzia bacteriană 0,5 ml și 0,1 ml. După 10 zi am avut că zona de inhibare a fost de 2 ml. Zonele de inhibare a creșterii bacteriene nu au fost stabilite, ceea ce ne permite să concluzionăm că este posibil să combine soluții de lucru ale bioregulatorilor cu suspenziile tulpine bacteriene. S-a observat că preparatele tulpine entomopatogene și compuși biologic activ prezintă un efect protector și stimulator asupra plantelor. Vă mulțumesc pentru atenție.
Thank you. Questions, please?
Spuneți, vă rog, unde ați izolat tulpina?
Tulpina noi singur am izolat-o din insecte, din insectele de fluture alb american. A fost izolată. Am fost depus la colecție la Institutul de Microbiologie, tulpina.
Așa. Atunci, dar dacă e așa, atunci dar o să vă spun că dacă e depozitat în colecție, atunci când o prezentați trebuie să aibă și cifră.
Până ce l-am prezentat din cauza aceea că eu am dat depozit documentele la brevetare și oricum
N-are importanță.
N-are importanță, da? Numai din aceeași, da, numai din cauza aceasta, adică chiar acum recent și așa am înțeles că nu trebuie să fie publicație, așa în general.
Da, să nu publicat. Dumneavoastră, pur și simplu, fără ca să o depozitați, vă puteți publica.
Bine.
More questions? I have one. Have you tested your biopesticides in practice?
Da, eu am testat. Noi suntem deja noi avem proiectul pe 2 ani de zile. Anul acesta l-am testat cât la cartof, atât și la mere. Este un efect foarte bun, fiindcă era, știți că anul acesta a fost așa o secetă extraordinară și cu 40-50 de grade ajungem, dar oricum, roada noi, oricum rezultatele acum sunt, da, trebuie prelucrată, dar roada e foarte bună.
You should have mentioned that.
Da.
Is your biopreparation comparable to those which are already on the market? Are you better than what we can buy right now, or comparable, or worse?
Noi încercăm, dar eu știu dacă vă face.
Cât de comparabil sunteți cu biopreparate care sunt pe piață?
Eu cred, așa, oricum, cercetările noastre până acum în lume nu sunt. Și sunt deja rezultate foarte bune. Și adică eu n-am cu ce compara, pot compara numai anume cu niște cercetători din Rusia care sunt niște preparate și insecticid și împotriva la bolilor. Oricum, nu sunt, până acum nu sunt așa rezultate. Anume cu derivații paraaminobenzoici.
Okay, thank you. Looks like you can make a lot of money on that.
Mulțumim.
Okay. Our next speaker is Samoilova Anna. You're welcome. Sorry.
Dear colleagues, let me present you a report on the study of the effect of phages isolated from different sources against fire blight pathogen. This pathogen, Erwinia mellifera, is very important from the economical point of view. Here you can see a healthy plant, queen's plant, and queen's plant affected with Erwinia mellifera. It is clear that this pathogen causes severe losses of crop, and it is needed to elaborate as effective means of the control as it is possible. During 200 years since since people know this disease, a set of control measures have been elaborated. Among them, deletion of infected plants, of course, using of copper-containing preparations, using of antibiotics, using of antagonistic microorganisms, gene engineering methods for obtaining resistant plants, and bacteriophages, viruses of bacteria, specific bacterial viruses. Viruses have been discovered in 1917, but in 1928, Alexander Fleming discovered antibiotics, and phages were postponed for 100 years, at least from the point of view of the protection from bacterial diseases. But during 100 years, bacterial revealed resistance to antibiotics, and researchers reminded about bacteriophages. And during the last 20 years, active investigation of bacteriophages is noticed. Bacteriophages are viruses which consist as where genomic program is recorded on nucleic acid, double-stranded acid, DNA acid, single-stranded DNA acid, as well as single-stranded RNA and double-stranded RNA. As you can see, they are of different forms, not this classical form as we know about. We know this T4 or T7, you see them everywhere, I think, but these filamentous phages, these sarcophages, Erwinia mellifera is infected by Myovirida phages with contractile tail, with Cipavirida phages with non-contractile tail, and with Podovirida with short tail. For the purposes of protection from bacteriosis, it is very important to know the life cycle of phages. According to the life cycle, phages are divided into 3 groups: viral phages, which infect bacterial cells, construct phage particles and destroy bacterial cells and go out, release these bacteria and to infect the other cell. The lysogenic phages, which incorporate their genome in bacterial chromosome. And this bacteria carries genes of bacteriophages. And the bacteriophages give some properties to bacterial cells which they didn't have without bacteriophages. And these pseudo-lysogenic phages, which exists in the bacterial cell in the episome. They don't integrate in bacterial chromosome, but they're inside of the cell, and they don't destroy bacterial cell until they feel that this cell is in a relatively safety. When they feel that this cell is in danger, so it has to be destroyed, they immediately leave these cells.
Oh, clever.
Yes, they're very clever. And the main advantages of phages, which can be used in the application against bacterial diseases, are that they are obligate parasites of bacteria, and a very important moment, they are natural habitants of the ecosystems. Actually, bacteriophages of fire blight of Erwinia amylovora are under the investigations almost since their first discovery, since 1921. And as I already told, that phages which infect Erwinia amylovora are from family Myoviridae. These are our pictures, which we made together with our specialist in electron microscopy, Dmitry Chertak. Yes, I have to mention him. And this is Myovirida with contractile tail. And this is a Sipavirida with non-contractile tail. The ways which can improve, which can increase phage efficacy against plant bacterial, not only plant bacterial, but in this case, we're talking about plant bacterial diseases. First of all, we have to use the only virulent phages which destroy cells and which don't give their genomes to the bacteria. It is better to use phages which were isolated from the places of their natural presence. And before using phages in the field, so to say, it is better to investigate this system in vivo in laboratory. And in case of Erwinia amylovora, we have such instrument, such tool, because it has been shown that during infection of the immature peer tissues, Erwinia amylovora use about 394 genes, and it has been confirmed that infection of immature peer tissues required the major pathogenity factors of the bacterium. That is why these model immature peer tissues are very comfortable to check the biological efficacy of phages against this pathogen. Here you can see ooze which Erwinia amylovora produces on the immature peer slice. So the aim of our work was to evaluate the ability of virulent Erwinia amylovora phages isolated from the aerial parts of the affected plants and from natural water reservoirs near fruits orchards or wild results trees to inhibit Erwinia amylovora growth in the immature peer tissues. Why we have taken water? Because in most cases it is comfortable to use preparation on the basis of phages diluted in water. For example, this agri phage preparation, which is registered in the USA, is recommended to dilute in water. We used bacteria Erwinia amylovora. We used two strains of Erwinia amylovora. This is a strain which we have isolated from queen's tissues. You can see the colonies of this pathogen on selective and semi-selective mediums. And we used for the second strain Erwinia amylovora was CFBP 1430, which was isolated in 1972 from Hawthorne, and which is well known, well studied. Its genome was sequenced. the phages which we used in this study, phages from quiz, which we isolated in 2013 from Hawthorne, and two phage isolates were isolated from water in Switzerland. This was near Zurich. The third one was isolated from near Zurich Lake, and the fourth was isolated from the water on the bank of Lugano Lake. These phages were investigated with the major capsid protein based PCR assay, and we have found out that first three phages are from the Myoviridae family, and the fourth phage is from the Podoviridae. The first strain we investigated with electron microscopy, and we used it in our experiment in our experimental Queen's Orchard to check phage effectiveness against fire blight. Here you can see symptoms which our tested microorganisms caused on the immature pear fruits. First one is these slices were infected with Erwinia mellivora only. These slices were infected with Erwinia mellivora and then treated with bacteriophages. But this picture was taken after 7 days after inoculation. And this is control. These slices were infected, were treated with the distilled water. What we can see on the after this experiment, slices which were infected with strain C.F.B.P. 1430 demonstrated symptoms of fire blight infection on the second day after inoculation. Moldovan strain caused ooze after 24 hours, in 24 hours after infection. Phages from Queens were the most effective because the symptoms of light, very light symptoms of very small drops of ooze appeared only on the 8th day of the experiment. And the most weak, but we think that it was also very good strain. Strain number 3, phages isolated from the pond near apple orchard. The symptoms, very light symptoms, were noticed, were registered on the 6th day of the experiment. So, as in conclusion, we can say that it has been demonstrated that bacteriophages from water revealed high efficacy against bacteria Erwinia mellivora, and all studied phage isolates successfully inhibited fire blight agent growth in the plant host tissues for about 7 days. And according to our experiment, treatment with bacteriophages against Erwinia mellivora should be carried out weekly if environmental conditions are favorable for the development of fire blight pathogen. Thank you for your attention.
Thank you. Questions, please?
Are there special motivation needed for bacterial phage, or you use bacterial culture for cultivation?
Of course, it is better to use this medium which is used for bacterial cultivation. In our experiment, we used LB medium.
and you have electronic microscope.
No. Electronic microscope. We
Not
have
yet.
Dmitry Dmitryevich Tertyak.
Cultivation.
Mr. Киселица, please.
Да, только если можно на русском. Да.
Да.
Да. Скажите, пожалуйста, насколько действие вот этих фазов, оно продолжительное? То есть вы можете сохранить их активность на длительное время?
В смысле, в культуре или в поле?
Ну, хотя бы в культуре, да, вот, чтобы
В культуре, да, они в хлориде
Если
Цельсия.
они просто холодильники и все, да?
В хлориде Цельсия они хорошо хранятся. Ну, в поле они же подвержены воздействию, во-первых, ультрафиолетовых лучей, которые губительны для всех вирусов. Потом там другие микро... Ну, в воде они хорошо сохраняются. Вот что удивительно, они в воде хорошо сохраняются. Это радует.
И сколько примерно вот самое больше вы мониторили, и они активны?
Ну,
Сколько
вот
времени?
в поле мы, кстати, вот этот вот сад, что я показывала в начале, вот эту вот айву, мы ее обрабатывали, ну там, я не знаю, там, наверное, какие-то благоприятные условия сложились, что мы можем сказать, что все-таки эффект бактериофагов, то есть, по крайней мере, на них не было симптомов прям таких вот страшных. Но лучше всего, даже вот по этим грушам, вообще по нашему опыту, при благоприятных условиях лучше всего обрабатывать каждые 7 дней. Если вот в течение 7 дней высокая влажность и высокая температура.
Actually, I wanted to ask a question related to this one. That the fact that the infection came back 8 days later in the best case, is it because, why? Because it looks like the more time, the more phages should develop there. And so the effect should be increasing with time. Why is it disappearing?
It was limited amount of bacteria and limited amount of phages. From the very beginning, we infected bacteria and treated with. We didn't add phages or bacteria. We have a limited amount and phages, and it was very favorable conditions for bacteria and phages, but PS slices the limited nutrient substances, and due to this, the amount of bacteria and amount of phages, it decreased. And the other moment, phages infect actively growing bacterial cells. When bacterial culture is adult, goes to, yes, when it's adult, phages are not, they are able to infect them, but not very intensively.
So I'm trying to understand how could bacteria outlive phages? Because in the beginning they were suppressed by phages, but in the end the phages were out of the picture, but while bacteria started growing.
Maybe we used not enough amount of this ratio of bacteria and bacteriophages. In this experiment, we used 10 for I don't remember how it is in English. 10 четвертые.
10 to the power of
10 to 4. Colony forming units of bacteria and 10 to 7 plaque forming units of bacteriophages. Maybe in these very favorable conditions, very favorable for bacteria conditions, we had to use some more bacteriophages, but actually in the nature, it is enough, this 10 and 7.
Okay. Thank you. And from the practical point of view, how efficient are phages? How cost effective are they?
From the economic point of view, actually it is not very difficult to multiply to get this preparation. You just need a set of very expensive equipment, but you need it for to buy once and then you use this equipment. And medium, they are not very expensive. All people who work with phages, and I think I can agree with them, they all say that it is easy to get these phage preparations.
Okay, thank you. Any more questions? Thank you very much. The next speaker to be invited is Dr. Smishna Ina. Слушайте, пожалуйста. Не надо.
So, the name of my report is Identification of Mixed Microbial Consortia Isolated from the Polyethylene Film Surface. And it's not the first report, and I'm not the first to report that polyethylene is a high recalcitrant and inert synthetic material and very difficult to degrade in the environment, and became a major source of environment pollution. The traditional methods for plastic waste disposal are recycling, incineration, and landfilling, the oldest and the most common method of plastic waste disposal. Natural degradation of low-density polyethylene, LDPE or PUP, depends on polymer properties, higher molecular weight, hydrophobic nature, and lack of functional groups recognized by microbial enzymes, and make it adverse towards degradation. However, several studies have reported the polyethylene degrading microorganisms isolated from the waste disposal sites. Various species of bacteria, such Bacillus or Pseudomonas or Brevibacterium or fungi, Aspergillus, Penicillium, Trichoderma, Andazes, or microbial communities isolated from soil are able to modify and consume the plastic polymers as a source of energy. So the purpose of present study is to characterize microbial consortia isolated from the surface of LDPE films extracted from the soil contaminated with polyethylene. So LDPE films were placed in the soil that was collected from the landfill located near the village of Slobodze Dushka, Kriolin district, the Republic of Moldova. And the soil with LDPE was treated under anaerobic or facultative anaerobic conditions within 6 months. Fungal and bacterial strains were isolated from the LDPE surface through enrichment techniques. At the initial stage, in the culture media was added LDPE, and as a growth inducer was added glucose. Samples from the enrichment culture were plated onto MSM mineral salt medium agar, nutrient agar, chapic medium. And grown colonies were selected and isolates were examined for the gram reaction, endospore formation, cultural characteristics such as color, colony form, margin surface, and elevation. And also studied the number of microorganisms in colony forming units, and after the 100 days of cultivation, the microorganisms in the consortia retain their viability, the titer being from 10 and 8 million colony forming units per milliliter to up to 32 millions CFU per milliliter. So they're viable. And you can see the colonies of fungi isolated from the LDP surface, grown on MSM agar. A total of 9 genera of Micromacetes and 3 genera of bacteria were isolated, described, and determined. So we see the Trichoderma, Fusarium, Penicillium, and also Penicillium. So as reported previously in the scientific literature. The isolated Micromacetes belong to different genera, 3 species of the genus Trichoderma, 4 species of the genus Penicillium, and 1 representative of the phylum Ascomycota. And the light microscope images of fungi isolated from the LDP surface is used for determination to the genera, to the species. So the consortia was described and composed predominantly of fungal strains, and Micromacetes are mostly represented by genus Trichoderma and representative of phylum or division Ascomycota, the main part of it. The first consortium of Trichoderma, Penicillium, and Ascomycota, and the second also 2 strains of Trichoderma, Penicillium, Ascomycota. The bacteria were determined only in 1 consortia out of 5 obtained, and were represented by the species from the genera Bacillus and Streptomyces, the 1 representative. And the last 2 consortia consist of 2 representatives of the genus Trichoderma and Penicillium, where Trichoderma predominated. It's the first stage of characterize the consortia, and the second stage is now under the investigation. So this is all founded by the project Microbial Tools for Degradation of Non-Recyclable Plastic Waste. Thank you for your attention.
Thank you. Time for questions.
So is a set report.
Spuneți, vă rog, când opiniile acestea active care deja le-ați izolat și le-ați obținut și să aveți rezultate foarte interesante, când o să fie depus în colecția națională?
Când noi determinăm activitatea lor și determinăm până la specie. Dacă ea nu este activă, foarte activă, nu trebuie să puneți la... Da. trebuie să ultimul an de cercetare până la identificarea activitatea și active, da. Și noi determinăm activă este lor în consorțium sau individual, da, asta e foarte important. Da.
Și cele care le-ați izolat și le-ați identificat ca active în proiectul trecut, când o să fie depozitare?
Cu pesticide?
Da.
Scuzați. Da.
Are you satisfied? Thank you. Now let me invite Pastalaki Olga.
Dear colleagues, today I would like to present to your attention the results of our research on the effect of fertilizer on the appearance of microorganisms during soil remediation. The soil pollution is a global problem which during the years of industrialization and modernization has turned into a threat for the health of the population and biodiversity and for the food security in the world. According to the data presented annually by the Ministry of the Environment, the problem of former pesticide warehouses and the adjacent lands in the Republic of Moldova remains a priority. In most cases, these warehouses are demolished or partially demolished, thus being a continuous source for environmental pollution. Soil pollution with pulp residues has devastating effect on the soil microbiome. Therefore, a common practice in bioremediation techniques is the use of organic or mineral fertilizers, which act as a trigger for the metabolic processes of microorganisms and respectively stimulate their growth. So the aim of our research was to evaluate the effect of fertilizer on the abundance of microorganisms during soil remediation. The object of study was polluted soil collected from the territory of the former pesticide storage located near the village Slobozia Dushka. Again, the bioremediation was established in plastic jars, each containing 1 kilogram of contaminated soil. As a control, the contaminated soil without remediation was used. The experiment was designed in oxic and cycled anoxic conditions. Under oxic conditions, the soil humidity was kept constant at 60%. Anaerobic conditions were created by saturating the contaminated soil with water, 80% for the water holding capacity. The transition from the anoxic to oxic conditions phase was repeated 5 times. In this way, 4 experimental variants were established to stimulate the indigenous soil microflora. Different amounts of fertilizer were added in 2 experimental variants, 3 and 4 variants. The dynamics of the microbial population in the soil was evaluated by spread plate method on nutrient media, considered the most informative for the comparative study of microorganisms involved in nitrogen transformation. Here you can see the dominant groups of microorganisms during bioremediation. It was ammonifying bacteria, which assimilate minor forms of azote, and oligoneutrophilic bacteria. Unfortunately, micromicetes and bacteria from the genus Azotobacter have disappeared after second and third cycle. After calculation of total number of microorganisms, we observed that most actively the microbial population increased in variant 4 compared to polluted soil. In this, the variant population increased by 155 times, and compared to variant 1 oxic conditions by 25 times. Remediation by cycled oxic and anoxic conditions, its variant 2, did not have big effect on microorganism populations, but the addition of fertilizers, variant 3 and variant 4, under the same conditions, significantly stimulated the abundance of the microbial population. Thus, we can conclude that the fertilizer used by us can serve as a bioremediation factor in cyclic anoxic oxygen conditions, which stimulates the development of the microbial community in the soil contaminated with the POPs complex. The optimal concentration of fertilizer for the growth of microorganisms under the conditions of our experiment was 6%. Thank you for attention.
Thank you. Questions, please? What was the fertilizer in variant 4? Was it mineral?
No, it's the same fertilizer, but a different concentration.
Okay, but I just missed, was it just mineral or you had some organic substance?
The mineral and organic, it's cum spune, wood. Rumbeguș de lemn.
Okay, thank you. Well, you speak about the abundance, but you measure that abundance on Petri dish plates.
Yes.
And it is known that on Petri dish plates grow less than 1% of microorganisms that exist in soil. Have you tried to measure total biomass?
Not.
Or
In this
activity?
case, not. And our experiment was concentrat asupra bacteriilor care asimilează azotul și pe care noi putem să le vedem, să le izolăm. Adică am limitat.
And why this limitation? Why are
Because not all microorganisms can grow on the media.
Oh, but I think the bacteria that are involved in nitrogen cycle are important by themselves. You probably had some connections to that. Yes. Okay, thank you. Are there more questions? Thank you very much. Our next speaker is Bogdan Golub-Nina. Is she present? If you are, yes, please. Okay. Can you share your screen?
Yes, sure. Tell me if you see this.
Yes, so far so good.
Okay, so thank you.
Can you please start the presentation mode?
Like this? Oh, one minute. Like this is going?
No, it's not the presentation mode.
One more time. You have not seen, yeah?
It doesn't work, but I don't see you trying the presentation mode. Look at that post sign in the right corner at the bottom.
Yes, I understand. One more time. Like this.
Okay, and now press the sign in the right bottom corner that goes for the
It's gone?
mode. All right, don't worry, just present your material.
I wanted to show, please, one more time.
No, don't do it. You don't do it the right way. So just continue.
Okay. So I, my apologize that it doesn't sync. I hope it doesn't work, yes?
It does. We can, at least we can see everything and we can hear you. So you can
Okay,
continue.
okay, I will start. So dear colleagues, I apologize if anything I can show, but I will tell about result. So we result about the ability and stability of bacillus, of some strains of Bacillus pseudomonas and lactic acid bacteria after 15 years of storage in the National Collection of Non-Pathogenic Microorganisms. The study material consisted of 20 bacterial strains. Conservation of scientific and industrial valuable strains of microorganisms for long-term storage without changing in morphological, cultural, and biochemical characteristics presented the main aim for every collection. Species like Pseudomonas and Bacillus are known for its antimicrobial activity, stimulator effect on plant productivity, producers of exametabolites for plant protection, and lactic acid bacteria, how we know, are important for dairy products obtaining. Our study, we start from reactivation of bacteria culture after lyophilization, and we used method of microscopy, assessment of bacterial viability, viable cell count by different methods, and also we studied technological properties of lactic acid bacteria, like organoleptic properties, acidity, lactic acid content, and coagulation activity. Cell morphology and colonial characteristics were observed on nutrient agar, and microscopic examination confirmed the purity of cultures. Lactic acid bacteria present Koch in short or long chains, were able to active coagulation, dense consistency, and without gas eruption. In the result of the investigation was determined that Bacillus strains and Pseudomonas are viable, and their titer was ranged from 6.8 to 7.6 decimal logarithm of colony, and for Bacillus, and to 7.9 to 8.1 decimal logarithm of colony for Pseudomonas. About lactic acid bacteria strains have titer ranged from 6.2 to 8.3 decimal logarithm for colony. Survival rate of those bacteria were minimum 78%, what we can see on what we could see at this picture. According to classification on the resistance against freeze-drying process, bacteria belong to Pseudomonas, Bacillus, Lactobacillus, Streptococcus genus, are medium and resistant to lyophilization process. Thus, the number of viable cells of all strains remaining in the ampoules are enough to maintain the culture viable. And here we can see data about time coagulation, about acidity, lactic acid in product after 20 and 72 hours. I highlight two strains without most moderate and high or maximum acidity. Obtained result correspond technical requirements for lactic acid bacteria. From conclusion, we want to say that this work confirmed effectiveness of freeze-drying process and high ability of strains to regeneration. After 15 years of storage, the ability of strains decreased only by 19%, but the titer of viable cells is maintained at sufficient level to preserve valuable properties. Thank you for attention.
Thank you very much. Questions, please? I see no questions. Thank you again.
Thank you.
Our next speaker is Kadrian Olivia. Okay, he's not ready. Then let me invite Yushin Nikita. Nikita, are you ready?
Hello, dear colleagues. Yes. Are you hear me?
Yes, yes, we can hear you. And we
And
can see
do you
your
see
screen.
my presentation?
Yes, if you can, please turn the presentation more. Yeah, thank you.
So, good afternoon, dear participants. The
And-
topic of my presentation is metal removal from erbium-containing wastewater using atmospheric potencies.
I'm sorry, can you turn your video on?
Yes.
So that we can
Second.
see you.
Sorry.
Your camera.
Yeah, I trying to find it.
Yeah,
Now I find it.
Thank you.
Yeah. So erbium is a rare earth element which has critical importance for industry. And today erbium compounds are used in metallurgy as additive for laser production, in nuclear technology to prepare reactor control rods. And in view of high commercial value of erbium, it's important to recover it from secondary resources. And several methods are traditionally used for erbium recovery, such as solvent extraction, ion exchange, etc. However, these techniques have limitation. They require a large volume of solvents and other chemicals. They have high energy consumption and generate a large amount of waste. And for the use of biological objects for erbium absorption and has attracted interest in recent years. The aim of this presentation of this study was to explore bioabsorption potential of cyanobacteria Afrosphaera potensis for erbium removal. In our experiments, we took 20 milliliters of erbium solution with concentration 10 milligrams per liter, and 100 milligram of Afrosphaera potensis biomass was added and then rotated off shaker for 30 minutes with 200 rounds per minute. And after that, biomass was filtrated and concentration of erbium in solution was determined by optic emission spectrometry, which you can see behind me. The effect of pH on erbium removal by biomass was studied at pH range from 2 to 6, and experiments were performed at pH values were not performed at pH values higher than 6 due to the formation of erbium hydroxide, and the maximum removal of erbium 70% took place at pH 3. The lowest removal of erbium was 41% at pH 2. At time experiments, the removal rate was very rapid. In the first 3 minutes of sorbent-sorbate interaction, 74 of erbium was removed from solution. Increase of contact time did not significantly influence the rate of erbium removal, and equilibrium was attained. In kinetic studies, good correlation between experimental and calculated amounts of absorbed metal was obtained for both models. Based on their coefficients, the correlation value by absorption of erbium on biomass can be better described using the pseudo-first order kinetic model. The pseudo-first model assumes that the rate of sorption sites filling is proportional to the number of unoccupied sites. The Freundlich model better represents the data for erbium biosorption, the coefficients of determination being higher than for Langmuir model, and this tendency may be attributed to the heterogeneous distribution of sorption sites on the surface of spirulina, since the Freundlich model assumes that sorption occurs on a heterogeneous surface by monolayer absorption, and the maximum sorption capacity calculated from Langmuir model was 30 milligram per gram, and B value indicated the high affinity of erbium to spirulina biomass. And temperature can directly affect the biosorption capacity of spirulina biomass. The experiments were performed in the temperature range from 20 to 50. The temperature almost did not affect the rate of erbium removal. An increase of temperature results in the decrease of spirulina biosorption capacity by 7% from 68 to 61. In order to evaluate the thermodynamics of bioabsorption process, the following parameters such Gibbs free energy, enthalpy, and entropy were estimated. Obtained free energy values pointed at spontaneous nature of bioabsorption process. According to literature data, enthalpy in the range from 2 to 21 kilojoule per mole is an indicator of a physical absorption process, and the negative charge of enthalpy indicated an exothermic process, while its value showed that erbium bioabsorption on Aphrosphaera potensis was physical absorption. And positive entropy values indicated an increase in randomness at the solid solution interface during absorption. And the biomass was dependent on several parameters, and the maximum biomass absorption capacity of 30 milligram per liter was achieved at pH 3 and temperature 20 degrees Celsius and 3 minutes of contact time. Freundlich isotherm showed its applicability for description of equilibrium data, while the kinetics of absorption was better present by the pseudo-first order model. From thermodynamic point of view, the bioabsorption process was spontaneous and exothermic, and the Aphrosphaera potensis can be considered as a safe and efficient bioremediator of erbium-contaminated environment. Thank you for your attention.
Thank you. Other questions? Looks like with time you will have a lot of biomass of spirulina with zinc and erbium. Are there any ways to dispose that biomass, or maybe even to obtain the elements from it?
Not now, but we want to do it in future. Like now we are finding the better parameter for bioabsorption. So, and we like make research if we can use spirulina biomass for remediation with elements, and after that we will try to obtain metal from this waste. Yeah.
Okay, thank you. More questions? Thank you very much.
Thank you.
Livio, are you ready? Livio Cadriano is our next speaker.
Это, да. I present the chromium bioaccumulation potential of a dark cyanobacterium Nostochlinia ground on multimetallic systems. Heavy metal poisoning in the exposure to heavy metals like lead, mercury, arsenic, and cadmium. Many of the heavy metals, such as zinc, copper, chromium, iron, and magnesium, are essential to body function in very small amounts. But if these metals accumulate in the body in concentration sufficient to cause poisoning, the serious damage may occur. Bioremediation is gradually becoming a standard practice for the restoration of heavy metal contaminated areas, since it is more eco-friendly and cost-effective compared to the conventional chemical and physical methods. Microorganisms such as fungi, yeast, and bacteria have been considered to be outsiding organisms for detoxification of pollution. Microorganisms are nutritionally flexible and able to adapt to extreme environmental conditions. They also possess numerous intracellular and extracellular enzymes which utilize the complex pollution and convert them into carbon and energy source. The microorganism can be involved in heavy metal uptake by employing two processes: intracellular accumulation, that is living biomass, and extracellular binding, third both living and dead biomass. Nostochlinia is a filamentous nitrogen-fixing cyanobacterium that is commercially produced by several companies meant for use as biofertilizer. Another use of nostoc biomass is to obtain polysaccharide, with therapeutic value, phycobilin, and antioxidant. As a result of this technology, a biological mass is obtained that can be used in various areas, for example, to remove heavy metal ions from the aquatic environment. The aim of this study was to assess the potential of cyanobacterium Nostochlinia to accumulate hexavalent chromium during 3 successive cultivation cycles on multimetallic systems. Object of study: cyanobacterial strain Nostochlinia deposited in the National Collection of Nanopathogenic Microorganisms of the Republic of Moldova. Potassium dichromate was used as a source of hexavalent chromium metal ions. Metal were added to medium in the form of salts. Metal concentration in the system are shown in table 1. The accumulation of metal in Nostochlinia biomass was assessed using neutron activation analysis performed at the Joint Institute of Nuclear Research, Dubna, Russia. The amount of nostoc biomass accumulating during 3 vital cycles was shown in figure 1. The first vital cycle in all polymetallic systems was characterized by a high level of biomass productivity. In the second cycle, nostoc showed a more pronounced sensitivity. The amount of biomass decreased by about 13% compared to control. The third cultivation cycle, as we can see, the biomass was at the level of cycle 2 in the case of the first two metal systems, while on the other 2 systems, the amount of biomass decreased by about 2 times compared to control. The rate of chromium uptake by nostoc biomass was very similar in chrome, iron, chrome, chrome iron nickel and chrome iron nickel zinc copper systems and varied with the cultivation cycle. In the three systems, chromium recovery in the first cycle was up to 40% of the initial level of metal. In the second cycle, up to 33%, and in the third cycle of up to 27%. In the chrome iron nickel zinc system, the bioaccumulation capacity of nostoc biomass was high and amount to 63%, 56%, and 35% of chrome will correspond of 2-3 cultivation cycle. Iron, nickel, zinc, and copper were also taken up during the cultivation of nostoc biomass from one cycle to another. In conclusion, the ability of cyanobacterium nostoc linker to bioaccumulate in chromium from the contaminated medium remained high during 3 generation, will the uptake of iron, nickel, copper, and zinc in the biomass increase from generation to generation. Research data have been published in the International Journal Environment, Volume 9, 2022. Thank you.
Thank you. Questions? I have a question. So many co-workers of yours use spirulina. Why did you go to nostoc? Do you have anything against spirulina? Toți în laboratorul vostru utilizează spirulina. De ce nostoc în cazul dat?
El e foarte promițător, dar merită să fii studiat.
Ați cumpărat cu spirulina?
Deocamdată încă nu. Pe viitor.
Well, it's good. It's good to hear that not spirulina alone.
Nu, că s-au mai fost cercetări și pe altul, nu numai spirulina. E așa, simplu la conferința aceasta e mai mult spirulină, dar așa se lucrează mai mult.
Yeah, I'm speaking about the impression, of course.
Dar așa, nu să-i spun domnul raportor, că mult a dezmerdat diferit colecția nutrițională de microorganisme nepatogene, dar nanopatogenă nu nimic.
Da. Bine, merci. Thank you very much. If there are no more questions, our next speaker is Makar Vasile. Okay, then let me invite for the second time Dr. Desnik Evelina.
My name is Evelina Lesnik. I will present a common work performed in our department of Physiology at the State University of Medicine and Pharmacy. This is more public report. It's from our side is more sample, and the interesting is about the video observed treatment in the management of patients of tuberculosis. So the title of the report is Efficacy and Safety of the Video Observed Treatment in the Management of Patients with Pulmonary Tuberculosis. So in 2000, in the Republic of Moldova was approved the first national program for the control of tuberculosis, which led to the national implementation of the strategy, which is named new as directly observed treatment short course chemotherapy. So we call this DOT. What means DOT? Which means that we have to observe directly the patient when he's taking the drugs. According to the updated national policy, the patients can benefit actually from the following treatment options. First of all is community or home-based DOT, when the treatment is delivered in the community close to the patient's home or work. This means that there is a special social trained health worker who goes to the patient's home and every day and gives him the drugs each day and is observing how the patient is following the drugs. Other possibility, which is most common, that the patient comes every day at the family doctor and under the supervision of the assistant is taking the drugs. The second option is the DOT is administered in the specialist healthcare hospitals or services. For example, actually we are speaking about the clinical hospital of pneumophysiology that is localized on the Strada Grenoble, which pose a lot of problems for Chisinau. The third, which is the most new one, is the video observed treatment, which can be synchronous or asynchronous based on the principle that the staff, trained staff involved in the performing of the treatment can observe the administration of the treatment using the electronic devices. And the most common is the cell phone, as I have there, rarely using the personal computer and other devices which has Android system. It's important to have a web camera. So it's important to have a broad internet, an electronic device connected to a specialized VOT platform, and of course the trained physician or medical staff. The advantages of video-assisted treatment that VOT can of course replace the DOT, which means directly observed treatment, when video communication technology is available for the entire population, or at least for the people which are enrolled in this type of treatment, and the healthcare provider and the partners are well trained in delivering this type of treatment. It allows evaluation of the adherence, so the compliance of the patients from the distance, avoiding the direct contact of the patient with the healthcare worker, by this way diminishing the risk of the nosocomial infection or interinfection. VOT is more flexible for patients and healthcare workers, achieves a higher level of interaction between patients and staff, and probably has a lower cost than DOT, if you not count a lot of cost of IT technology. So the aim of the study was to assess the efficacy and safety of VOT in patients treated with pulmonary tuberculosis. The first objective was, as usual, assessing the peculiarity and risk factors of patients treated via VOT or DOT, compared to DOT. And the second is to establish the efficacy and safety, and to identify some recommendations. So in general, the patients underweight the VOT is in a very low number, because there are a lot of criteria which should patients encounter to be enrolled for VOT. And this study group was compared with a group of patients which were treated standardized via DOTs, which means via directly observed treatment. So what we can identify is that for DOT, the rate of men were much more higher than in VOT. So the general mass of patients with tuberculosis is composed in 2/3 of the proportion by men. However, in VOT, we cannot identify such a big proportion. Distributing patients in age groups according to the according to the WHO, we can establish that even if it was not identified a statistical difference between groups, however, the very young patients aged less than 25 predominated in the group of VOT. And distributing all groups in two types, less than 45 or more than 45 years old, we received the statistical predominance of young patients aged less than 45 in the group treated using the video assistance technology. It's important to know that we identified a high difference between the distribution of patients from demographic areas. So it's important to know that wide-band internet was more accessible to the urban patients, and that's why the majority enrolled in VOT, of course, were residing in urban sectors of Chisinau. And also it's very important to emphasize that the patients who were declared themselves or they could not provide a residence, which means carte de identitate with visa, they were approximately each 7 patients in treated under DOT. I have to emphasize that patients without a stable place of residence usually are treated in the hospitals till the end of the treatment. So such condition could not be included in the group of VOT, so that's why there is a statistical difference between the groups. It's important to emphasize about the peculiarities and risk factors which encounter the patients to achieve these types of treatment. So low socio-economical state where we include unemployed, students and retired statistically predominated in the group treated classically using the DOT. Lack of insurance, of course, which is associated with unemployment, first of all, as well predominated statistically in the group treated classically. We have to emphasize that lack of insurance is not a barrier to be hospitalized in the management of patients with tuberculosis. So even in actual practice, every second patient which is hospitalized is without insurance. So this characteristic, of course, predominated in the DOT group. Low educational level, which means lack of, in general, studies or primary or incomplete secondary school, predominated in the group treated classically, and higher level, which means at least bachelor or secondary school, professional school, and high school predominated in the VOT group. Also, we had some information about the matrimonial state, which is not so relevant, and we identified that probably the single persons predominated in the DOT group. However, this is personal information, and we could not check or establish correctly. We have such characteristic as poor living conditions, which is very subjective in our conditions, because most of the patients with TB are living in poor conditions. However, we could give this characteristic to those without residence of living. So because we could not check for other groups. And of course, we received statistical predomination of patients living in poor conditions within those treated classically, and that's why they opted to receive the treatment in hospital conditions where they have food, place of live, and etc. So about the homeless and lack of stable residence, of course, we said that this is a condition that excludes the VOT. And also it's important to emphasize that migration or recent returning from abroad predominated in the VOT. Here I should emphasize that in general, every 10th patient with tuberculosis in our country is recently returned from abroad. Those are economical migrants, and due to exposure to very severe conditions, social conditions, high stress, they are returning with active processes due to immunosuppression associated with low social state in the hosting country. Other conditions which exclude the possibility to enroll patients in VOT is the history of detention, because most of them, the patients who were incarcerated, they usually no place of living, and alcohol abuse. Alcohol abuse is associated with psychic impairment, and this is the first contraindication to enroll the patient in new treatment. Active tobacco smoking is absolutely associated with tuberculosis in absolute majority of cases. So even in pediatric population, teenagers, I can say. So of course we could not assess very in a high proportion. However, because the patient will not say if the patient is smoking or abusing alcohol, so you can only check the behavior or specific smell. So for patients treated classically, each predominated compared with those treated via VOT. A very low number of drug users were identified in the DOT group, and it's important to emphasize that patients from TB clusters, so which have also family members with TB, they opt for VOT treatment. So normally in previous experience, we were taking the patient from the TB cluster, and we were proposing the hospitalization to exclude the contact. But the actual tendency, epidemiological tendency is that the patients from TB clusters to opt the VOT and to maintain their household, because every patient is a member of a family, so has duties. So this is something new for us. And associated diseases, in general, patients with TB, even they have some wrong behaviors, they are not so burdened by associated disease in general. So the rate of patients with associated diseases was around 30%. This is a general rule. And HIV infection represents exclusion of the patient from the VOT, and because they are considered very immunosuppressed patients that should be hospitalized and monitored very strongly. So those patients were enrolled only in the DOT group. So in general, the treatment outcome also is very interesting as the results. So we identify a high rate of treatment success. So for the VOT, we have 87%, and for DOT, we have 76%. This is a very good success in general for this period. So patients were treated in 2000. So this is the group of 2000. Because before, in the previous years, we had a very low treatment success. But due to the fact that we are implementing different approaches, which is more close to the patients, the rate of treatment success is very high, and rate of drop up or lost follow-up or abandon is low. Because healthcare staff is highly involved in the recovery of the patient. So the patient with TB cannot disappear from Moldova. So anyway, they will be included in the treatment. And it's important to know that treatment failure was identified, of course, but the rate was very, very low. And also, we have this situation, the patient lost from follow-up, which means the patient who doesn't want to continue the treatment for more than 2 months, or the patient who has some contraindication and the healthcare staff finish the treatment. So this is the general definition of the patients lost from follow-up. So we can establish that even in VOT, when all the conditions are applied to the patients, the loss from follow-up cannot be avoided. So this is something usual. So we cannot neglect this situation. And also the deaths cannot be avoided, neither in the VOT group and of course in DOT group. So even for VOT were excluded patients with severe social peculiarities, 3 patients died due to associated diseases. So anyway, this approach doesn't avoid loss to follow-up and the death. For DOT, of course, this is patients treated classically, of course, in hospital, and every 10th died from TB. This is the general rate, and of course the majority due to progression of TB, and the minor proportion due to associated diseases. Here I have some practical aspects. We have this platform TB-Vote.MD. So we have the doctor's name who should log in, and of course the patient should log in through the patient's name. Here it's describes the institution who will, yes, excuse me, this is asynchronous VOT. What means asynchronous? Means that the patient will take the drugs in front of the camera, of the cell, as mine, Xiaomi 6, and is recording, and the recording is uploaded in this application, and the doctor, logged as the doctor, will check the correctness of the swallowing and will validate this video. So here you have the institution. Here is written that there is a new video that should be checked. This is asynchronous VOT. Here one aspect of registration of the patient. The doctor or assistant special trained should stay 10 minutes and checking the video. Okay, then the medical assistant should record herself, explaining to the patient everything is good or is not good, how is better to improve, and to validate, or and to ask if the patient during this video will say something. So the will ask advices. The medical staff should explain. So then video is confirmed, that was checked. The video answer of the medical staff is uploaded, and here you have the location. What's important to emphasize that this possibility of the treatment is prohibited to be done outside the Republic of Moldova. So normally the staff should check in which country is located the patient. Other advantages that we have a panel which expose the names of the patients that underwent the VOT. The patients will can of course report the adverse drug reactions. Here we'll have also the entire list of the adherence by date and month of the patients attributed to each doctor, which will show the days in which the recording were uploaded and the days of the accepted recording. So everything is entirely monitored. And we, of course, we can extract the Excel file with those patients. So I finished quickly. VOT video-assisted treatment represents a modality for the anti-TB treatment delivery when certain conditions such as electronic device and the wideband internet are put at the disposition of the patient. So this project, which gave to the patients the possibility to use the VOT, they included in these expenses the phone, abonnement with internet traffic included, the management of the applications. So this project has a special staff which is involved in the uploading the application, registration of the patient, and the registration of the doctor who will check the patient. So there is an intermediate organization. The advantages of the VOT is that it facilitates the treatment follow-up correctly, but does not replace DOT when patients have important contraindications, deep social vulnerability, such as homeless. We know because the patient is, if the patient is homeless, we will not be able to get him when he will drop out of the treatment, which means we need a special location where the patient can be found. And of course, we need a person who will be the backup, which means a family member or social member. So if we lose the patient, we have quickly to find this person who will find our patient. A mental impairment, so this is psychic diseases, so no one with psychic disease can be included in VOT, as well as those with very low educational level as well cannot be included. Alcohol abuse, this is clear, drug use, HIV infection. This is an important contraindication because it's associated with severe immunosuppression and severe diseases. So he could die at home if the healthcare will not take place very quickly. So this is an imminence of death, and that's why it's an exclusion from VOT. And of course, of the location, the patient could not be located abroad from Moldova. This is excessive from my point of view, because especially for those working, for migrants who are working outside of Moldova, they should be involved in VOT. However, there is another barrier because the patients will not receive medication every day. So they have to go to the specialized healthcare and to take the box of the drugs for 1 week. So if the patient will be located outside, he will not be able to come to take the drugs. But in general, for example, for other countries where there is no barriers in transportation, of course, it could be used. So the challenging impact of the complex risk factors on the treatment outcome did not differ significantly in patients treated remotely compared to the patients treated under direct supervision, which means that loss to follow-up and the death cannot be prevented in the group of the patients treated via VOT. So this is normal. So it should be accepted as it is. So in general, the VOT in patients with pulmonary TB showed a high efficacy and safety regardless of the treatment phase during which was implemented, which is about that before was this project adopted to those patients who could be treated completely in the intensive phase and continuation phase via VOT. Now they observe that it's possible to treat during the intensive phase in the hospital, and when they go to the continuation phase and he should go to the family medicine and to take the drugs, the patient could be transferred in VOT. So actually we make the diversification of the implementation of the VOT. So disease-related characteristics didn't show any impact on the treatment outcome. And the rate of adverse drug events is low. I didn't know we didn't present it here, but in general is very low because in this type of treatment, the patients are included which has no important risk factors which could determine the adverse drug events. And there's a recommendation in general that VOT can be implemented at the national level in actual epidemiological conditions if there are supporting measures for patients to be implemented. And thank you, over.
Thank you. Questions? Thank you very much. Just one remark, included, not included. So is Makar Vasily available?
Yes.
Yeah, you're invited, you're welcome. Yes, yes, please come here.
Bună ziua la toți membrii. Eu aș dori să mulțumesc din partea colegilor la Universitatea Tehnică de posibilitatea de a participa la simpozionul dumneavoastră. Îmi cer scuze, nu am avut lecții, de asta am fost nevoit să venim repede din deal la Petricani. Lucrarea prezentată de noi este efectul produsului zoobioar asupra sănătății, metabolismului mineral și producției de ouă la găini ouătoare. Am concretizat în prima perioadă tehnologică de ouă. Sunt mai mulți colegi aici, n-am să dau citirii. Scopul nostru a fost de a investiga produsul nou asupra păsărilor, asupra sănătății, productivităților și în special care este toleranța acestui produs din partea găinilor ouătoare. Nu știm foarte bine că păsările se exploatează acum în condiții intensive de fabrică agricolă, sunt supuse stresului tehnologic care influențează atât sănătatea cât și calitatea produsului. Prin urmare, noi am urmărit posibilitatea utilizării acestui produs și impactul lui asupra a mai multor parametri, inclusiv în caz de față a statului clinico-hematologic, metabolism mineral și producției de ouă și unor parametri morfometrici ai ouălor de găină. Despre produsul dumneavoastră o parte știți foarte bine, este obținut în cadrul institutului dumneavoastră. Produsul este în condiția mea, în condiții care nu le cunoaștem, inociv, nu are sau nu conține erbicide, nu are toxine, nu conține conservanți. Noi l-am folosit mai just un an jumătate în urmă, acum am inițiat altă cercetare pe prepeliță și până ce noi suntem mulțumiți de prestarea acestui produs și a noastră. Iată, produsul care poate nu cunoaște. Ce este important pentru noi? Că acest produs conține microelemente ca zincul și seleniu, elemente necesare pentru fortificarea sănătății, statului imun și în special acelui antioxidant. Așa. Cercetările s-au făcut într-o fabrică de păsări la Floreni, pe 70 de găini ouătoare, care au fost la vârsta de 129 de zile. Mai jos vom vedea loturile. Am avut în total 5 loturi, a cât de 14 păsări. Lotul martor, după cum vedem, n-a fost supus unor intervenții, rația a fost cea stabilită și aprobată la fabrica respectivă, iar 4 loturi au inclus în rație a câte 5 miligrame kilogram furaj, 10, 15, respectiv 20. Mai departe merge lotul experimental 1, 2 și 3. Păsările au fost permanent monitorizate, periodic cântărite. Ouăle erau colectate individual pe loturi, separat. S-a determinat evident temperatura corporală, mișcările respiratorii. Într-un context general, și arătăm că au fost investigate mai multe substraturi biologice, sângele, ficatul, țesutul muscular și ouăle. În cazul de față, mai mult noi vorbim despre sânge și ouă. Noi am recoltat sânge de 3 ori. La debutul studiului, la circa o lună de zile de la inițierea cercetării și la finele studiului, la 4 luni de la debutul studiului. Sângele s-a recoltat din vena axială, iar la sfârșit păsările au fost sacrificate inițial, totuși sânge s-a recoltat din venele jugulare. Ulterior, păsările au fost evident sacrificate și recoltate și alte substraturi biologice. O atenție deosebită a fost acordată de noi în cazul acestui studiu parametrilor metabolism mineral, calciu, fosfor, ferului, ceruloplasminii și transferinii și a fost calculat din matematică și raportul calciu-fosfor. Cercetările noastre au arătat pe durata întreagă că nicio pasăre practic nu a fost care să dea reacții adverse la produs, eu mai știu, aparatul tracto-gastrointestinal sau alte reacții, să zicem, vomă la pasăre, mai știu ce puteți să fie, sau diaree, adică reacții absolut n-au fost, mai mult ca atât. Preparatul a manifestat unele reacțiuni benefice, care permit să vorbim și despre o adaptare mai bună celor din loturi experimentale. În baza datelor s-a constatat, e clar că ar fi inutil să citesc toate cifrele, a constatat că temperatura corporală a fost mai mică la loturi experimentale, cât și mișcările respiratorii, care sunt niște date confirmate și prin investigații anterioare de noi și permit să vorbim despre o acțiune benefică. La nivel de sânge s-a constatat o acțiune pozitivă asupra gamma-hematopoiezii. Dacă ne uităm la prima vedere, pare că eritrocitele manifestă o slabă tendință de diminuare și este un fenomen constatat de noi și în alte studii la păsările oătoare. Ulterior, noi vedem că volumul eritrocitar mediu este mai mic, volumul eritrocitar. Este un lucru foarte bun, fiindcă dacă eritrocitele au volum mai mare, cum vedem la lotul martor, deci vedeți, 124 kilolitri, atunci dincolo ce avem 120, 121, indicatori care vorbesc despre o posibilitate mai bună de deplasare a eritrocitelor prin capilare. Și totodată se permite să evidențiem lotul 4. Vedeți? Negătând că este un preparat natural, ecologic, dar vedem că totuși doza mai mare are unele momente, să zicem, care permit așa mai simplu să evidențiem o doză mai bună în acest domeniu. Concentrația ericitrară medie a fost la lotul experimental 3 mai bună, iar volumul, sau mai jos concentrația, acolo volumul, deci concentrația eritrocitelor în hemoglobină a fost mai mare la lotul experimental 2 față de alte loturi experimentale și mai jos vom vedea și alți parametri. Ce se referă la metabolism mineral. Evident, calciul este un element bine cunoscut și el este un indicator, un parametru relativ indicatori buni și în lotul martor, însă la cele experimentale se vede o tendință de creștere, în special la lotul experimental 2 și 3, pe când la finele studiului, refer la fosfor, parametrul este mai mic la 3 loturi experimentale, 1 2 și 3. Și apare întrebare, de ce fosforul e mai mic? E mai bine? E mai bine. Și lucrul ăsta vorbește nu că nu este fosfor, dar că este o utilizare mai intensă a fosforului care în tandem cu calciul participă la crearea cojei de ou și la alte procese. Lucrul ăsta se confirmă și prin raportul calciu-fosfor, care este mai benefic, după cum vedem la loturile experimentale. Și în picioare iarăși 1, 2 și 3. Iar la lotul 4, cum am spus, doza, să zicem așa, cea mai mare, maximală, este mai mic parametrul la fine față chiar și de lotul martor. Referitor la concentrația ferului, datele care prezentate primiți, așa, concluzionăm că totuși remediul testat influențează metabolismul ferului atât în generarea ferului necesar, cât și utilizarea lui pentru procesele metabolice. Așa, scuze. Nu, aici de acum este redacția ce am menționat și mai sus în unele cifre. Referitor la producția de ouă. Noi ce am făcut? Zilnic am spus că s-au colectat ouăle, iar scoatem în evidență aici debutul studiului, când în fiecare lot, practic, vedeți, erau câte 10, 9 ouă. Diferența nu era. Ceea ce permis vorbim despre sănătatea păsărilor, corectitudinea studiului și altele. Pe parcurs, intensitatea ouatului, iată, vedem, la 10-a zi a fost de 109 ouă, așa e producția numerică mai just. Loturile experimentale au fost parametri mai mari. Ce este important? Că la sfârșitul studiului, parametrii investigați au fost mai buni, cu circa 100-150 ouă față de cel martor. Intensitatea de ouați, iată, în loturile experimentale a fost 100%, în lotul martor 91,7 aproximativ. Viabilitatea păsărilor, adică integritatea lotului, n-am avut păsări moarte. Vedeți, unde au fost dozele mai mici și lucrul ăsta s-au constatat și în alte situații. 100% în celelalte loturi am avut decese la păsări moarte. Deci e reflectat și în procentele mortalității. Așa. În paralel cu cele menționate, vedeți, sunt cifre, dar iarăși n-ar vrea să reținem atenția. Noi, când am terminat studiul, am hotărât să investigăm ouăle. Prin investigare, ce e posibil în Moldova. Practic, nicăieri nu se fac analizele, deci biochimici ceva ouălor. Și noi am investigat parametrii morfometrici. Și mai jos vom vedea. Și am procedat așa. În primele 2 zile după colectarea ouălor, la 2 săptămâni și la 1 lună. Și sunt iarăși parametri extrem de mulți și cifre de s-au luat în calcul. Masa ouălui, diametru mare, diametru mic, lungimea gălbenușului, înălțimea mai jos, lățimea, masa lui, înălțimea albușului, masa albușului, greutatea cojei, etc. Și s-a mai determinat evident și grosimea cojei, de la capătul mijlociu, rotund, și ce s-a scos în evidență. Adică așa, aici la 2 săptămâni, iarăși un lucru enorm, și atât doamna doctorandă, dar asta nu e teza ei, teza ei se începe acum prepeliți materialul, a făcut cu mâna ei tot lucrul acesta și vreau să mă opresc la 1 lună de zile. Și la 1 lună de zile au fost niște rezultate bune care au scos în evidență că greutatea ouălui s-a menținut mai mult la laboratori experimentale și într-un context legat cu siguranța alimentelor, iată, grosimea albușului a fost mai mică ca la lotul martor, iar înălțimea gălbenușului și greutatea mai mare, ceea ce permite să vorbim că procesele așa, să zicem, negative, care pot avea cu timpul sau care ouălor a fost mai mică și protejată de remediul testat de noi, care este un lucru foarte bun. Ca exemplu, vedeți, la lotul experimental 2, înălțimea albușului este cea mai mică. Așa, e de 0,84 cm sau cu circa 6 cm față de lotul martor, pe când greutatea albușului este cu, vedeți, 1,5 55% mai mare față de valori lotului macro. Incontestabil, aceste date vorbesc despre calitatea oului la un termen de 1 zile de păstrare, date care au semnificație mare în siguranța alimentelor. Așa, nu, aici în așa fugitiv, păsările au fost cântărite și s-a demonstrat că la finele studiului, sporul total perioadă, adică a fost pozitiv, dinamica la toate loturile, însă la lotul martor, dacă a fost de 59 de grame, apoi la loturile experimentale, deci 1, 127, 128, 125 grame. Aceste date vorbesc încă o dată despre studiul corect realizat, despre sănătatea păsărilor și alte, necătând că în condiții tehnologice, chiar dacă când găinile au o perioadă intensă de oat, putea să fie chiar și puțin scădere în greutate. În cazul de față, cum vedem, dinamica a fost alta. Ce putem menționa? Că rezultatele obținute permit să constatăm că remediul n-a influențat negativ starea de sănătate, dozele administrate de 5, 10, 15 mg au indus valori apropiate ale masei corporale la găini, iar doza de preparat 20 mg substanță activă furaj la kilograme de a influențat mai puțin greutatea păsărilor, nu la mare. Date care permit să scoatem în evidență doza optimală. Aici iarăși, în concluzie, produsul, perioada de 121 zile s-a administrat zilnic, n-a influențat sănătatea și a fost bine tolerat de păsări, a influențat o acțiune benefică asupra sănătății, în special asupra statului clinico-hematologic, cât și rezistenței naturale. S-a stabilit că la loturile experimentale de păsări tratate cu zoobiuri, indicatorii de bază a producției de ouă, precum numărul de ouă, intensitate, sunt mai buni în comparație cu astfel de parametri la lotul X-martor. Iarăși, cu referire la calciu, s-a indicat o superioritate a acestui element la loturile experimentale și o diminuare a fosforului față de lotul martor. Aici se evidențiază în special lotul experimental 2. Mai departe, se menționează concluzii despre diminuarea erului, cum am menționat, și ca recomandare, ce noi putem menționa? Indicatorii biochimici, zootehnici obținuți. Permis să propunem administrarea produsului bioactiv, zoobior, găinile ouătoare tinere, produs care se poate suplenta hrana în prima perioadă tehnologică de oat. E un termen de circa 4 luni de zile. Și vreau să vă spun că în paralel cu cercetarea asta, noi am avut altă cercetare pe găini bătrâni, zicem, în ultima perioadă. Și acolo indicatorii au fost mai puțin evidențiați față de aceasta. De atâta este o dovadă și prin cercetări și prin alt studiu în paralel că la debutul studiului preparatul intervine ca un adaptogen, fiindcă este o perioadă stresantă începutul oatului și ar fi bine folosit numai să fie și putem să-l implementăm. Vă mulțumesc pentru atenție.
Vă mulțumim și noi. Întrebări?
Domnul profesor, spuneți, dumneavoastră ați menționat că preparatul influențează pozitiv asupra parametrilor belgărușului.
Da.
Asta e evident, cum ați spus, e siguranța alimentelor, e un plus foarte mare. Spuneți, domnul, ar însemna aceasta că aceste ouă sunt mai bine venite sau mai, cum se găsească cuvântul, pentru a obține urmari din ele? Adică pentru...
Da, dar din păcate e clar că așa cercetări nu s-au făcut și ar fi... Nu, asta așa nu mă putem să intuim, chiar și într-un context științific sunt mai bune. Dar iată, dumneavoastră a spus tot de calitate. Ce vreau să vă spun? Chiar undeva ce ne-au ținut teza de master și am confruntat. Și pe urmă am mai avut cândva și un seminar și tot am discutat. Cred că e timpul noi să trecem de la noțiunea asta de eficacitate economică la calitatea produselor. Când credeți-mă, des să folosesc produse mai puțin calitative, sintetice. Și e clar că forțează sănătatea păsărilor, dar ce obținem noi, din păcate, puțin asta interesează. De atâta, în contextul menționat și chiar și în direcția care este la noi în stat, prin intermediul Agenției Naționale pentru Siguranța Alimentelor este o direcție bună și merită să luăm în calcul cercetările și produsele care noi va să le obțineți.
Mai sunt întrebări?
Deci, rog, sporul greutate la lucrurile
Experimentale.
experimentale era mare, era comparativ cu mare. Asta nu poate fi deja obezitate.
Nu, găina dacă ar avea o 4-5 kg, da, dar acolo ce 100 era, dacă la lotul martor era 59 de grame, așa, și dincolo 120, diferența, să zicem, de 60 de grame sau plus minus, nici de cum.
Asta e sporul în greutate pe toată perioada.
Da, pe toate. Asta nu zilnic. Zilnic nici n-am mai stat acolo, undeva iese un gram jumate, 2 grame.
Dar ficatul. Am văzut că ați făcut și pe ficat.
Da. Dar eu am arătat numai de ce am vrut să accentuez, fiindcă totuși mulțumim și doamnei director, fiindcă ne ajută preparatul să înțeleagă că noi lucrăm serios în acest domeniu și nu. Dar ficatul, noi avem cercetări, sunt și publicații care demonstrează sănătatea ficatului, fortificarea lui și așa mai departe. Și mai mult ca atât, iată, recent a ieșit o publicație în Brazilia și aici am implicat niște colegi din România, s-a demonstrat și la nivel de intestinul subțire și așa mai departe, că mai bine se adoptează după parametri acolo, histomorfologi și așa mai departe. Și o lucrare așa, de categoria IC, care recent.
Mersi. Mai am și eu o întrebare, dacă se poate. De obicei, preparațiile în baza spirulinii sunt destul de costisitoare,
Așa.
chiar și pentru oameni. Dumneavoastră aveți 130 aproape de zile cu administrare zilnică și întrebarea mea este dacă nu se primește ou de aur aici.
Da, nu, cam de argint ies așa. Dar uitați-vă, noi am luat așa. Dacă într-un mililitru de substanță sunt 20 de miligrame substanță activă, așa? Ieșa la, să zicem, la 1 kilogram, 1 mililitru, așa? Dacă lotul 4, celălalt revenea de mâncare și pasărea mănâncă în jur de 100 ceva de grame, adică pentru cercetare a fost bine. Și chiar și să le folosim preparatele la oameni, trebuie să le trecem prin animale. Și eu am încercat produsul ăsta și el e bun și ajută, vă spun.
Așa, avem o dovadă vie atunci. Mersi mult. Mai sunt întrebări? Atunci, we have our last speaker, Moldovan Ana.
Cum să încerc să-ți fac?
Puteți chiar să faceți. Some problems with the disk.
It's okay, you just open it.
No, it says it has problems. You have a different disk?
You can scan it, I think.
Is it yours?
Yeah.
Which one? Which one? Which
So this
one?
presentation will the one at all. This one. This one, yeah. Dear colleagues, dear organizing committee, dear scientific committee, first of all, I would like to thank you very much for this great opportunity, this framework to meet both physically and online with all of you. And for me, it's a great pleasure to, first of all, get knowledge about the latest developments in all in our country and in international background or in different areas of biotechnology.
And thank you for this opportunity to present my speech about local strain of Bavaria bassiana and our attempts to go beyond to local production of biopesticides, and a first step in identifying the best culture media for growth and sporulation. The problems residing from this topic is that chemical pesticides are a huge amount used in the agriculture. You can see that in 2020 in EU, it was used over 346 thousands of tons of chemical pesticides, even though we know that these pesticides are associated with a huge number of problems, and our farmers need these tools that have to come from the biotechnology. So we have a huge amount of strains that can be isolated from the natural environment, but now they are not used and developed at a great extent, but they can be used both in industry, in agriculture, and for environmental protection purposes. And we have isolated the local strain, and we have proved that it is good to be used as a bioactive agent for local preparations of biopesticides. And we have tested it against several of the pests, and it showed promising results. So we wanted to know which would be the best nutrient medium for the cultivation, so as to be used for medium scale cultivation. And we have started with our strain grown on PDA medium. And after that, we have transferred mycelial discs to different culture media prepared both in the laboratory or bought commercially. We have counted for the number of conidia produced and for the germination rate. So in this table, you can see the media used. We have tried to reach the media that we can take from the market, from different producers. Let's say one of the most often used media for the cultivation, and if we are searching in the web, the articles, we will see two medias for the Bavaria bassiana. It's PDA, so potato dextrosis agar, and SDA, subbord dextrosis agar. So we tried to reach it from our Republic of Moldova market, and we bought PDA from Merck and High Media, and the SDA from High Media. And also we have bought a selective agar for pathogenic fungi from Merck. We know that it will inhibit the growth of non-pathogenic fungi, and it's used like a selective media for Bavaria bassiana, and we used this as control to compare with that. And searching the literature, we also have seen many works speaking about the fact that it's good sometimes to add yeast to the SDA so as to have the improvement of the growth and sporulation. So we have tested this media. And also we have tried PDA with yeast, and we have tried to produce PDA in the laboratory from the standard TCP, and also to substitute the glucose with sucrose. And we also have tried to prepare by ourselves a media, very simple media that we have, when we search literature, we have find that actually one of the first description of the Bavaria bassiana was actually made on the oatmeal agar. And after that, I don't know why, but it was somehow left behind, and everyone have switched to the PDA or SDA. And we have prepared it by ourselves. So we have transferred the mycelial discs by 5 millimeters in diameter central to the petri dishes, and we made 5 replicates for each medium used, and we have monitored daily the growth. And we have used statistical analysis for the data. So here you can see the vegetative growth of our strain on the 14 days of the cultivation on different media. And we have compared the data to different types of PDA. So PDA without any additional letters, it is from High Media, M it's from Merck, A it's from our own production in the laboratory, and PDA, it was the addition of the yeast. So we can see that comparatively, the produced by ourselves PDA and those that bought from Merck and from high media were not statistically different data regarding on the vegetative growth. So we can see that adding the yeasts after several days, let's say after 12 days, somehow our strain grow not so, so the speed of the growth is not so high as the others. So adding yeast is not improving the situation. And also we have calculated the mean gradual growth rate in millimeters per day. And you can see this data, so not statistically different between the media from commercially available or prepared by ourselves. So also we have tried different media. So we take as comparison the PDA produced by ourselves, SDA with yeast, you can see in this slide. You can see that what was expected, SAP, because it's a selective media, will inhibit the growth also of the Beauveria bassiana. The others were comparative, the same if we are analyzing radial growth. If we compare with the media produced by ourselves, we can see that the best radial growth was recorded for oatmeal agar. You can see in green dots this media. So, and the data are statistically significant, different. So, and here you can see the data for all the media that were tested. So we have the best mean radial growth rate on oatmeal agar. But we were interested not only by the speed of the growth, but also the quantity of the conidia that were produced per unit of surface, because we actually are interested in taking the conidia and using them as the part of the biopesticide. So we have quantified the number of conidia per square millimeter, and you can see the difference in these two pictures. So the growth rate and the conidia produced. The number of conidia per square millimeter produced were the highest when we are using PDA with addition of the yeast, but comparative the same as PDA from the high media, although the growth rate was lower on this media. And oatmeal agar actually, regarding the number of conidia, was not the best. But when we have calculated the total conidia per petri dish produced, because of the radial growth rate higher on the oatmeal agar, it has improved the data. So it is comparable with PDA and SDA, which are so popular in the literature. But beside the quantity of the conidia and the speed of the growth, we are also interested in the germinative rate of the strain after the cultivation on this media, because being a bioactive part of the biopesticide, we are interested how fast this will germinate after the cultivation, because the infectious process is dependent on the germination of the strain. When it is used as a biopesticide, we have to be sure that the strain will germinate and penetrate the cuticula of the insect. So we have tested which is the germination rate of the strain after the cultivation during 2 weeks on different culture media. So we have analyzed the data, and here we have seen something very interesting for us that we were not expecting to see. And we made 4 replicates using the medium. So we've seen the growth on SDA, but the germinative rate was very low. So when we had made the germination rate, we didn't see any attempts to germinate. And after that, on that plates, nothing developed. But we can see the oatmeal agar performed very well compared to with the rest of the medium. So SAP was, we were expecting that it will also inhibit the germination rate because it's a highly selective medium, and it also inhibits the entomopathogenic fungi a little bit, and it's used in the selective isolation. So here we have the synthesis of our results. If we consider all the viable conidia per plate produced, we can see that oatmeal agar has performed the best. We had the best results. And on the second place was PDA from high media, even though we were expecting that the Merck will somehow be better because we have worked a lot with, but high media also performed very well because differences is not so high, but when we are interested about a high amount of conidia, yeah, so these are not statistically different, yeah, between those two. So we have concluded that our strain can be successfully cultivated on ovarose solid media, both commercially available formulations and media prepared in the lab. The maximum radial grow rate was recorded on the oatmeal agar. Also, the highest number of viable conidia per total was produced on this medium. We can say that the fungi grow rate differs depending on the culture medium used, and the same culture medium, but from different producers, can distinct a different, let's say, pattern of the grow. Sometimes it grows faster, but the conidia produced are lower. And also we have underlined the necessity to check the purchased media and to identify a simple cost-efficient media that can be easily prepared on site. And also, if we want to go to the producing this, we have to set quality control points, because if something changes and we don't know the composition of the media, it can influence the germination of the strain. And thank you so much for your attention.
Thank you. Questions? I have a question.
Yeah.
What would you say is the most important scientific result of your work?
So if taken into account, we had an applicative purpose of our work. So if taking from the scientific point of view, I think that this work had proven the importance of preparatory phase before going to the producer. And also that in each producing line, we have to have a research facility which will investigate how the specific medium influences, and that we have to check for the growth and sporulation of these strains before going into producer, because we cannot simply take the data, let's say, and replicate, because it won't work. Every strain has its own particularities that have to be taken into account. So for us, it was like many of the papers were recommending SDA, but in our work we have shown that this medium was very bad in the germination. And we could not just take and replicate this and be sure that everything's okay and not check the control points. Because then we will say that our product is not working in the field, and it's because we haven't checked that it's not germinating. It grows very well, a lot of conidia produced, easily to formulate after that, but it's not germinating after this media.
Do you have any suggestions why? Because all those different media, they can help you by looking at the reaction. They can tell you something about your object of study. Did you come to any ideas or mechanism why germination is more active in one case and less active in the other?
So let's say we compared the medium with the same composition and source of carbohydrate. We changed the source of carbohydrate from a simple hexameric and to sucrose, which already is too, but we haven't checked, let's say, the composition of the oatmeal after boiling, so as we can relate the best result to the different oligosaccharides that are forming there. But sure, when it has different sources of energy, then it will enable different processes inside of that. But yeah, we can check. Sure, if we, let's say our next step will be we would like to set up solid state fermentation as a first step. That's why also we have introduced oatmeal, and we will test different sources of carbon for this.
Thank you.
Thank you for your question. Thank you.
Any more questions? Then thank you very much. And thank you very much for your attention. I guess our day one is over, isn't it? No?
One more, no?
Do we have any extra reports? I don't have anyone on the list. So it's over. Okay.
Cum credeți independența de mediu schimbă patogenitatea?
Este de asemenea un punct care este foarte important de a fi verificat, confirmarea virulenței. Deci, să zicem așa, o sursă de carbon nu ar putea să influențeze atât de mult virulența, deci, pentru că virulența de fapt este determinată de producția enzimelor care hidrolizează chitina de aceea noi oricum o să confirmăm păstrarea virulenței, da? Pentru mai mult virulența este afectată de cultivarea succesivă în mediile nutritive, pentru că se inhibă genele respective. Și atunci noi, pentru a menține în cultură această tulpină, noi o reactivăm, o trecem prin gazde, da. Dar vrem să încercăm, de exemplu, să vedem dacă ce ar fi dacă să nu trecem prin gazdă succesiv, pentru că e o problemă recuperarea după asta a tulpinii. Dar, de exemplu, să adăugăm insecte cadavre sterilizate și cumva să activăm enzimele respective. Noi am început deja să facem aceste cercetări.
Atunci trebuie uscate, făcute pudră și adăugate.
Da, în mediu pentru reactivarea virulenței, fără procesul nemijlocit activat de o gazdă care penetrează.
Asta e problema și pentru destructori de pesticide și alte destructori, da, trebuie să activează.
Noi o păstrăm tulpina primară, o păstrăm în glicerol și mai mult, deci alicote, dar pentru cercetările respective, deci în frigider și am văzut și acum noi suntem cu criza asta de lumină electrică, alergăm cu cutile pietre acasă și înapoi, tot nu e o soluție. Am găsit niște publicații din spațiul sovietic în care se realiza cultivarea pe medii, deci pe discuri deshidratate și la temperatura camerei și mulți ani, bine că nu pentru toate lucrează, dar o să încercăm, acum am setat o linie de testare ca să nu fim dependenți de cam așa, dar este o problemă în care noi toți acum existăm și mulțumesc foarte mult, mulțumesc conducerii institutului pentru această oportunitate frumoasă de a vedea.
Mulțumim și noi. And thank you for being thankful, by the way.
Yes, thank you so much.
Thank you very much for your attention. We will meet tomorrow at 8:30. You are very welcome. Until then, goodbye.
Картин, да. Мы câteva zile, cum imediat obținem. O să fie și firma o să vă invităm să luați chiar și cartea de aici. Noi imediat vom veni pe tipografie. Facem mai târziu, când modificări au fost în un moment.
Contactați colegii din colecție, încercați să vedeți cum funcționează păstrarea din colecție. Poate mai ușor. Poate mai ușor să le organizez și să o păstrez. Я так понимаю, что сегодня вы